everybody. Welcome to your lecture today. I want to take a minute and talk to you about graphing and some of the important aspects of it because, as you know, in science, it's all about the data, right? Whenever someone wants to quote you different aspects of something, the first thing that should pop into your mind is where's the data? Show me the data, right? So let's talk about data. Um, when you're actually performing an experiment, which you have read about inside your module with the scientific method, you're going to gather data and you're going to get all this data and you're going to have these different numbers. And usually, what you'll do is put it into an Excel spreadsheet, and it gives you all of these different options of different ways to craft your data. So what you're looking at here is a scatter plot with lines, and this is an experiment that was done a few semesters ago. And so using the scientific method, the question that was posed was, I wonder whether or not 66 students could actually meet each other in class within 5 minutes. And so we proposed the hypothesis that 66 students could meet each other within 5 minutes. And we ran the experiment, looked at all the variables, figured them all out, ran the experiment, and here's the data. So here on the y-axis, we have the number of students met, and here on the x, we have the number of students in class. And you can see that each student actually had their own data point as to the number of students that they met. And so this is a very interesting graph. This is one way to actually plot your data. But here's another way to plot the data. And it's a little bit more difficult to actually interpret what this graph is trying to tell you, right? When you look at this graph, what you see is the number met is here in orange, in this squiggly line. And here the number of students in class is shown as this line that's accelerating and maxing out at 66. Really hard to interpret this data. So there's different graphs that you can choose, the exact same data, and get two different graphs. And one, is kind of adequate. You'll see scatter plots shown a lot, sometimes with the lines, sometimes without the lines. But the way you actually choose to present your data makes a difference as to whether or not people are able to easily, at a glance, take a look at your data and understand what your graph means. And that's the whole point of it, right? You can never change the data because that's a big no-no. The data points are what the data points are. However, the way you choose to actually represent that data in a graph form is really up to you. So what I want to explain to you now is a type of graph that you'll see a lot. It's called the box and whisker plot, and I want to make sure that you understand what it means. So with a box and whisker plot, basically you're taking all your data and you're dividing it into 4 parts called quartiles. So here, this long whisker is the first quartile. This box is the second quartile. This long box here is the third quartile, and this long whisker here is the 4th quartile. Now, the end of this whisker basically tells you the minimum data point. That's the smallest data point, not including the outlier. This line tells you it's the median of the data. Please look up what median means. This X, if it's present, tells you that that is the mean of the data. Please look up what the mean means. And this end of the whisker tells you the maximum of the data. So you can see box and whisker plots that are actually lying horizontally, as you see here, or most of the time you'll see them in this vertical orientation. And basically it's telling you the exact same thing. You've got your maximum, the greatest value data point here at the end of this whisker, your minimum, least value excluding the outlier here at this whisker. This is your lowest quartile, the lowest 25%. Here's your median. This is where 50% of the data is greater than this value, the middle of the data set. You've got your upper quartile, the upper 25% of the data here, okay? But let's look at outlier, what that is. So we've got an upper outlier, so an outlier that was actually more than 3/2 times the upper quartile, and a outlier here at the bottom that's actually less than 3/2 times the lower quartile. So when we see these dots, that tells us these are the outliers. And what do outliers mean? How do we interpret outliers? Well, outliers can have different meaning, and when you get an outlier in your data, you need to investigate it to determine whether or not it's something that's very important or whether or not it was an error in the input of the data. So we don't just throw outliers away, we still present them here, as you see, as these dots. And most of the time what we'll do is explain them after we've investigated the outlier in the actual write-up itself. So if it turns out that it was an error in input, meaning that maybe someone wrote a 0 when they meant to write an 8, it was interpreted by the person who was putting it into Excel as a 0 when it actually was an 8. So it turns out that's an error. And so you can say that was an error, and so we omitted it. So here it is in its original form, and when we omitted it, this is what the data looks like because it was an error, right? Or most of the time, outliers actually aren't errors, human error in input. It turns out that they actually just occurred in the experiment. And so you need to take a look and determine whether or not that outlier is very important. So let's take a look here at the data that we gathered. So here we have now that exact same data, except it's represented in a box and whisker plot. And you can see that it's very easy to interpret this graph now. So here we have the number of students in blue in this box and whisker plot. We can see that the maximum number was 66. The number of student METs is represented here in this orange box and whisker plot, and we can see easily by looking here at our mean and our median that it was about 25. The highest number of student METs was about 35, but then we have something going on down here, this little dot right here. And this is actually our outlier, right? And so in this experiment, where we're trying to determine how if 66 students can meet 66 students in 5 minutes, and of course we adjusted it because you can't meet yourself, so it's 66, can 66 students meet 65 students in 5 minutes? That somebody met 0, right? When we look at that, that means that more than one, at least one, met 0 people, right? And it could be that maybe they meant to write an 8 and they wrote a 0, right? So we investigate the outlier, and it turns out that no, it actually was a 0, but it turns out that those individuals either had a cast on their leg and they couldn't get up and walk around to meet anybody else, and no one came over to meet them, which is interesting because that would spark a whole new experiment. Like, how do you meet people when you can't get up and move around, right? So a whole new experiment would be performed because of that outlier. It sparked a whole new thought process. Or what if this were a cancer data set, and what we were looking at were the treatments to our new experimental drug, and this here in the orange, instead of number of student MET, is actually the number of people who did not live. The cancer actually got them, right? They died. Except this outlier, this person actually survived. Now suddenly that outlier becomes the most important data point in that entire data set, right? And so you want to actually evaluate your outliers and determine what category are they in. Are they an error? And you need to basically state why moving forward they're not going to be shown in the data set because it was an error of input. or whether or not it's an actual data point that is true and factual. However, upon further investigation, there didn't seem to be any interesting findings that are derived thereof. So you still show it, but you explain it. Or it's not an error, and it actually is the most interesting data point in the entire data set, and here's why, right? And it's going to spark a whole new experiment, right? So don't just throw outliers out. We never ever get rid of data. Even if it's an outlier, we go on to try to explain it. So now you know how to read a box and whisker plot. You know how to read a scatter plot. You know how crafting your data in different forms of visualization, data visualization, can be very important as to the one that you choose so that people can actually take in the information in a one-shot fashion. By just looking at it, they can understand what you're trying to represent. But there's another way that you can represent data. And here we have a word cloud. And so say this actually was done, this experiment was run by a class, and everyone wrote down their major. We took all the data and we collated it together, and we can see that the majority of the individuals in class were business majors, followed by psychology, followed by media and communications, sociology, political science. And so here in the word cloud, the words that had the most data points actually were increased in font size. So at a glance, we can see which ones had the most individuals. So that's just a different way of representing data. And so data can be gathered in different ways. It can be represented in different ways, but you can never change the data that you gather, okay? Do you guys remember this dress? Ah, the dress. Oh, so old. Why the dress, right? It turns out that this dress is going to help us make a very good point. So when you look at this dress, either you see it as white and gold or you see it as black and blue, right? Big deal. Okay, there were huge fights on the internet as to whether it was black and gold or whether it was black and blue, white and gold, black and blue. You know, people were just like, no, no, no. But there were some experiments that were done around this, and it turns out that if you see the dress as white and gold, then you probably spend more of your time outside in the sunshine. And if you see the dress as black and blue, you probably spend most of your time indoors in artificial lighting, okay? And why is that important? Why do you care about that? Why was that experiment done? That experiment was done because if just by showing you an image, I know where you spend the majority of your time, that's a huge data set that you probably want to get your hands on if you're a marketer, right? Meaning if I know that you spend most of your time outside because you see this dress is white and gold, then I probably want to market my sunglasses to you, right? Because you're going to need them. You're spending most of your time outside. Or if you see this dress as black and blue, I suddenly know that you spend most of your time indoors, and maybe I want to market you my energy drinks because you're probably inside gaming the whole time, right? So a lot of data can actually, interesting data can be gathered in different ways. So you do want to be careful as to the amount of data that you give away online, because sometimes you may be giving away more information than you think you are, more than just the color of a dress. Okay. All right. Let's move along. The other thing that I want to point out here, let's see if we can get our video to work. Watch our happy little cat sitting here watching a motorcycle race on TV. Let's see what happens. Boom. That's nice. The power of the cat, right? Totally swiped that motorcycle and down he went. Let's watch it again. Yeah. Okay, so I show you that video to point out that correlation does not equal causation. Meaning that just because the cat swiping its paw on the motorcyclist did not cause the motorcyclist to fall, as you know, right? Seems very obvious in this video, but many times you're going to have correlations presented to you as causation, and correlation does not equal causation. And so you always want to look at the data. So whenever someone is telling you that something is caused by something else, you want to look at the data. Show me the data, right? So I can take a look. Show me the experiment where you actually had all these different cats basically swiping a screen at random times and making the motorcyclist fall. Show me that data, right? So it's all about the data, and correlation does not equal causation. All right, let's move along here. We're going to get to the meat of what we're dealing with today, which is the chemicals of life. All right, we're going to talk about atoms in chemistry. So, terminology, elements. Elements are substances that cannot be broken down chemically into other substances, such as gold, carbon, copper. These are all elements. When we look at basic atomic structure, because I know some of you have taken chemistry, some of you have not, we're going to start very basic so everyone's ramped up with the same information. The basic structure of an atom is you have protons, neutrons, and electrons. Protons are found in the nucleus of the atom. They have a positive charge. Electrons reside in shells circulating outside of the nucleus. They have a negative charge. And then neutrons are also inside the nucleus. Okay, so 3 basic parts of the atom: protons, neutrons, and electrons. Inside the nucleus, you have protons and neutrons. Protons are positive, neutrons, as in their name, they're neutral. They have no charge. Electrons are negative, and they reside in shells outside of the nucleus. Okay? So you've got forces of attraction that are happening between the positive charge of these protons in the nucleus and the negative charge of these electrons that are basically circulating in shells outside of the nucleus. And that's what's holding the atom together on a very basic level. So here we're looking at the hydrogen atom. We see it has 1 proton, it has 0 neutrons, and it has 1 electron. Here we're looking at the carbon atom. We see that it has 6 protons, it has 6 neutrons, and 6 electrons. And so there's actually something that you all know and maybe don't know how to interpret yet, called the periodic table, and it's the list of all the elements that we know of in the world, and it's actually being added to when new elements are found. And so I want to show you how to actually read the elements in their tabulated form here, because it's not as difficult as it looks. You look at it, it kind of looks like this complicated alphabet soup. It's actually not. It's going to boil down to each element has its own little card, if you will. It has a letter, or letters, plural, which connotate the name of the element here, H, which stands for hydrogen. It has a number up here and a number down here. We're going to look at what these numbers mean. Okay. When we look here at oxygen, again, O for oxygen, there's a number here at the top, and it's the atomic number, and that is the number of protons. And the way that I remember it's the number of protons is because the atomic number is the number at the top, which has a P. It's the number of protons. Okay. The number here at the bottom is the atomic mass, or the atomic weight, and it's the combination of the atoms, protons, and neutrons. Okay? So, now we know there's 3 basic things that make up an atom. We've got protons, neutrons, and electrons. When I look at the atomic number, the number at the top, the number of protons, I automatically know how many electrons are there in the atom. It's the same as this number, 8, because it's balanced. It's a balanced atom. It's stable. It has the same number of electrons as it does protons. okay? But when I want to know how many neutrons are there, all I have to do is take a look at this atomic mass number, which is protons plus neutrons, and subtract the protons, and then I know how many neutrons, right? That's just simple math. So if I start out here with the atomic mass of 15.9994, which is rounded to 15.9, and I'm trying to figure out how many neutrons are there, I basically just subtract the number of protons because I know that this atomic mass is protons plus the neutrons. So I subtract the protons and I get the neutrons, 7.99. Okay? Super simple. And that's really all this is telling you. That's what all of those cards have. They have the atomic number, which is the number of protons at the top. And then they have the atomic mass, which is the number of protons and neutrons. And they're arranged according to their different elemental behaviors. And that you will get into when you take chemistry itself. We won't get into that. All right, let's move along. So I told you that the electrons actually circulate in these shells. So I need you to remember the first two shells and how many electrons they can hold, because this is very important. So the first shell can hold a maximum of 2 electrons. Once 2 electrons are in that shell is filled, and all other electrons have to go into the next shell. And the second shell can hold 8 electrons. So if we look here at oxygen, we know that it has, let's look here at its number, it had an atomic number of 8. It's the number at the top, it's the number of protons, the number of electrons as well, we can deduce. So 8 electrons. So let's fill in 8 electrons. So we've got 2 in the first shell. First shell is now maxed out, 1, 2. We start putting the other electrons in the second shell. So that's 2, 3, 4, 5, 6, 7, 8. There's all 8 of our electrons, 2 in the first shell, and then we have 6 in the second shell. But we know that the second shell actually has 8 spaces. We only have 6 electrons. So that means that oxygen actually has a vacancy. It's got 2 vacancies, right? 2 places where its shell is not completely filled. That makes oxygen very reactive. When atoms take a look at their outermost shell, they always want the outermost shell filled. And this is how we know whether or not an atom is going to be reactive or not. If it has vacancies in its outermost shell, it's going to want to react with another atom because it wants that shell filled. Okay? It either wants to give away electrons in order to become completely stable, which means its outermost shell is now empty. Maybe it had 1 in its outermost shell and it would like to get rid of that 1. Or here in oxygen, it's got 2 vacancies, so it needs to react and fill those 2 vacancies. Let's look at how that happens. So here we're looking at unstable atoms. We have hydrogen. It only has 1 electron. It has a vacancy in its outermost shell because this first shell can hold 2. It only has 1, so it wants to react. It's unstable. It's a very reactive molecule. When we look at the element or atom of helium, we can see that it has 2 electrons. That means that this outermost shell is filled. It's a very stable atom. It does not want to react with another atom. When we look at nitrogen, we can see it has 1, 2, 3 vacancies in this outermost shell, which should be able to hold 8. It only has 1, 2, 3, 4 in its outermost shell. 1, 2, 3, 4, 5. It has 5 in its outermost shell because we can hold 8. So it's got 3 vacancies. It is unstable. It wants to react. When we look at neon, all of its outermost shell, it has 8 in its outermost shell, is filled. This is not a reactive molecule. It doesn't want to react with other molecules. Okay? So we can look at the outermost shell to tell whether or not a molecule will be reactive or not. But let's take a look at carbon and the versatility of carbon and why it's so versatile. It's so versatile because when we look at its outermost shell, which is the second shell here, it has 1, 2, 3, 4 vacancies because it only has 4 electrons in its outermost shell. So it's got 4 vacancies to fill in its little life, 4 holes in its little life that it wants to fill. And that makes it very versatile. It wants to bond with other molecules because of those vacancies. And so we can see that it will bond, carbon will bond with oxygen. We know that, and we actually create that bond inside ourselves to create carbon dioxide, which we breathe out each time we breathe out. Carbon is, in its different conformations, it will bond with oxygen and hydrogen in order to form sugars, such as glucose and fructose. And then it'll also bond with other molecules, such as nitrogen and oxygen and carbon, to form proteins. So carbon is extremely versatile and is extremely important to our life. We're even called carbon-based life forms, right? But when we look at the breakdown of elements that make up our body, we can see that oxygen, we're about, 65% oxygen, followed by carbon, hydrogen, and nitrogen. But these are all 4 main elements, right? Carbon, oxygen, hydrogen, and nitrogen. However, it's the conformation of those atoms that are used to form extremely important molecules. So for example, I can take a basic piece of paper and using origami, fold that paper into different conformations and make like an elephant, and then fold it again and make a bird. But it's the same sheet of paper, right? It's the same 4 elements, oxygen, carbon, hydrogen, and nitrogen. But how they're actually combined in that conformation actually is making different proteins, like the protein that makes your hair or the proteins that make up your antibodies. It's because the conformation of those atoms is different and it makes a totally different molecule, even though it's using the same base atoms, okay? So let's move along. So you've probably heard these terms before, isotopes and ions. Let's go ahead and define those. Isotopes are atoms with the same atomic number. Remember, that's the number at the top, the number of protons. So same atomic number, same number of protons, but the atomic mass is different. So if I have the same number of protons, but my atomic mass is different, what has changed? Well, the atomic mass is the combination of protons and neutrons. That means my neutrons are different, right? So here we look at carbon-12. It's got 6 protons, 6 neutrons, and 6 electrons. The atomic mass is 12, hence the name carbon-12. But when we look at carbon-13 and carbon-14, we can see that we have in carbon-13 7 neutrons, which makes our atomic mass 13, 6 protons and 8 neutrons for carbon-14, which makes our atomic mass 14. So isotopes, atoms with the same number of protons, but a different number of neutrons. And these are really important because we use carbon-14 to do carbon-14 dating, right? We can actually look at the breakdown. We know the breakdown of carbon-14 over time. And so we can measure the amount of carbon-14 that's in, say, a fossil, and then look at how that carbon-14 is actually degraded over time. And then we know the age of that fossil. So we use isotopes for dating different materials, but we also use isotopes in medicine. We can inject them, and as they break down, they release different forms of light that we can measure with different machinery, and we use that for diagnostics. So different isotopes can actually be injected in order to aid in medical diagnosis and diagnostics. And so here again, this slide is just pointing out to you that isotopes just have a different number of neutrons, okay? Protons are the same. Atomic mass is going to change because the neutrons are different. And atomic mass is just the number of protons plus the number of neutrons, okay? If you want to know more about carbon dating, because it actually is a very interesting subject, you can go here to this link by UPenn, University of Pennsylvania, and they have a really interesting page all about carbon dating and sort of that timeline of breakdown of that isotope. Okay, but let's talk about what ions are. So you're very familiar with salt, and salt is sodium chloride. But let's look at how sodium chloride actually is formed, because here we have the element, the atom of sodium, and we have the atom of chloride. Why do these two bond together? Why is it such a common bond? So here, when we look at the outermost electron shell of sodium, it has one electron. It really would like to get rid of that one electron so it can become more stable, right? When we look at the outermost shell of chloride, it is almost filled, except it has one vacancy. And it would really like someone to donate an electron to fill that vacancy. And along comes sodium, and sodium will gladly donate an electron. Now here's the thing. When sodium donates an electron to chloride, suddenly sodium is going to become more positive, because remember, we had a balance of electrons and protons that are in the atom. When we give away an electron, I'm going to have more protons. That atom's going to be more positive. When I give that electron away, the atom that takes that extra electron is now going to become more negative. And that's exactly what ions are. So when an atom has a more positive charge, it's called a positive ion. And when it has a more negative charge, because it actually gained an electron, it's called a negative ion. All right? And so these two want to bond together. All right. So different ways that these stick models, or the models of atoms, can be shown. One, we have the Lewis model that you probably have seen and you're very familiar with. Then we have the ball and stick model. And then we have the space filling model. So these are just different ways of basically showing you the same thing. So don't let the change in the visuals throw you off. It's just showing the same thing. Let's take a closer look at bonds that I need you to know and their strength. So the first bond we're going to look at is called the covalent bond. And in the covalent bond, remember the co means together, like co-working. You work together with that person. So lean to your Greek and your Latin to help you understand what these terms mean as we get more into scientific terminology, that these prefixes will actually help you understand and remember what the terms mean. So covalent bonds means that they're actually sharing an electron. They're sharing an electron together. So here we have hydrogen. Remember, it has one electron, and it also has one empty space in this outer shell. So two hydrogen atoms would like to bond together. And when they do, they're actually sharing that one electron that they each have, so that they both now feel like they have two electrons in their outermost shell, right? That's called a covalent bond. A covalent bond is a very strong bond. We can see the same thing with oxygen. So we can see the two oxygen molecules are covalently bonded. We can see here that they're sharing these two pairs of electrons are shared between the two oxygen atoms. Also, we can see that when we look at other molecules and their covalent bonding, we can see that carbon can covalently bond. Here we have carbon sharing its electrons with hydrogen, and it's actually 4 hydrogens here, because remember, carbon had 4 spaces, 4 open vacancies in its outer shell. Hydrogen had 1 electron, and so they are covalently sharing the electrons in order to become more stable. So different atoms can actually covalently bond together. And I just want to remind you that the covalent bond is a strong bond. So the thing that I need you to remember are the types of bonds. We're going to talk about 3 types of bonds, and the bond strength. So a covalent bond is a strong bond. When we talk about ionic bonds, remember, this is sort of the opposite attract principle that's happening here. When we have our ions that are being created, our example was sodium and chloride, making sodium chloride. So that's your ionic bond. So we have the covalent bond and we have the ionic bond. And remember, the ionic bond is a strong bond. Okay, covalent bond is a strong bond. The ionic bond is a strong bond. And when we look at the ionic bond, or when we think about the ionic bond, the molecule that we think about the most is the one that we use every day and that our bodies need, which is sodium chloride. And when we get this covalent bonding with sodium and chloride, it makes that very familiar crystalline structure that we call salt. But I want to take a look at some other features. So I told you that the ionic bond was actually a strong bond, but you would probably think, well, wait a minute. If I have a glass of water and I put some salt in the water, the salt dissolves. So how can it be a strong bond when I can put it in water and it completely breaks the bond? Let's take a look at that. So water has a very unique property. It has what's called polarity. So if you look at the image here at the top right, we can see that our oxygen, because it has so many protons, when it's sharing, covalently sharing, the electron with the hydrogens, which only has one proton, the electrons are actually spending most of its time with oxygen. And that's going to give the oxygen more of a negative charge. And at the same time, because the electrons are spending more of their time with oxygen, that's going to give the hydrogens that are there more of a positive charge. And so that gives this molecule of oxygen polarity, right? And that's really important because it gives water very unique properties. So let's take a look at how salt is actually dissolved in water on the molecular level. So here we have our sodium and chloride. They were bonded together, but what happens is the chloride, which has a more negative charge, remember it's an ion, because basically it actually obtained the electron from the sodium. So it gave it a more negative charge. Because of that negative charge, the hydrogens in water are going to be attracted to it. Because remember, because of the polarity of water, the hydrogens have a more positive charge. So they begin to surround that chloride. And you can see here in this illustration that the negative charge chloride now is surrounded by the hydrogens in the water molecule. At the same time, the sodium, which has a more positive charge, because remember it wanted to get rid of its electron, now it has a more positive charge, and it begins to be surrounded by the oxygen side of the water molecule, which has a more negative charge because of the polarity of water. And so as the water begins to surround these atoms of sodium and chloride, it's pulling them apart. And that's how salt is dissolved in water. All right, I want to carry on and take a look at the last bond that I need you to know at this point, and that's the hydrogen bond. So what's the hydrogen bond? So in a water molecule, oxygen's 8 positively charged protons attract electrons more readily than does the single proton in the hydrogen atom. And as a result, the electrons are pulled towards the oxygen side of the molecule, making it slightly negative in charge, while the hydrogen side is slightly positive. So we saw that, right? And so they're sharing the electrons, right? These are the covalent bonds between the oxygen and the two hydrogens, right? And that gives us the polarity of water. But it turns out that when you have these molecules of water, the H2O, they actually begin to have an attraction between each molecule. Because remember, because of that polarity, the oxygen has a more negative charge, and now the hydrogens in another water molecule are actually attracted to that negative charge. And so we start to see the polarity causing hydrogen bonding between the oxygen of one water molecule and the hydrogen of another water molecule. And you would think, okay, so why does that matter? Well, it actually ends up giving water some very unique properties. So this hydrogen bond that forms, hydrogen bonds are actually a weak bond. Remember, our covalent bond was a strong bond, our ionic bond was a strong bond, but the hydrogen bond is actually a weak bond. So the attraction between the slightly positively charged hydrogen atom of one molecule and the slightly negatively charged atom of another water molecule causes this hydrogen bonding, and it's a weak bond. But let's take a look at some of the unique properties of water that you're very familiar with, but let's look at why. So I want to introduce you to this little fella. This is the Jesus Christ lizard, and I'll show you why he has that name.
Some young Costa Rican basilisk lizards are looking for food. Basilisks have been nicknamed the Jesus Christ lizard. Why? This adult male, probably the father of the young lizards, can't tell us. Nor can this female, exclusive property of the territorial male. It has nothing to do with the feeding habits of the basilisk, which consists primarily of insects and berries. This predatory reptile will help reveal the secret as it stalks the young basilisk. The basilisk is called the Jesus Christ lizard because it can walk, well, really run on water.
Pretty neat, right? So you also are probably familiar if you've ever been to a river or a lake or a stream, where the water is very calm. You will see insects actually walking on the water. So not only can some insects walk on water, but also some large animals, organisms can walk on water, such as this lizard. But let's look at why they can do that. So it turns out those hydrogen bonds that we talked about between the different water molecules actually end up forming a net. And this is called surface tension. And so water actually has a high surface tension. And so certain insects and also lizard, as we saw, can actually walk on this surface tension of water. We, as humans, cannot. Be nice if we could. But let's answer the question, which I posed at your introduction video. I said we were going to talk about how water actually gets to the top of a 100-foot tree or even a 300-foot tree without a pump. Let's look at how that happens. So it turns out that this one property of water, which is called cohesiveness, together, two water molecules together, because of the cohesive properties of water, trees such as this giant sequoia that you see here in the image are able to transport water molecules from the soil, down where the roots are, all the way up to the leaves at the very, very top. And here's how that happens. So the leaves, the underside of the leaves of plants, they have an opening called a stomata that they can open and close, much like a mouth, right? Hence the name stomata. And when it's open, then water can evaporate out from the leaf, right? And when that happens, because these water molecules are actually hydrogen bonded together, it's much like a little daisy chain. I don't know if you ever, when you grew up, you had those little toys that you can just stick together and then pull them apart. It's kind of the same thing when it comes to the hydrogen bonding of water. So as one water molecule actually evaporates out from the leaf, it's pulling up the next water molecule in the daisy chain. So it's as if one of the students actually gave a really good illustration that I think would probably help you. It's as if a bunch of people were holding hands to make a chain. And as one person took a step to the left, it dragged everyone one step to the left. That is the way the cohesiveness of water actually works in bringing water up from the roots to a plant. So as one water molecule evaporates and leaves, it pulls up the next water molecule. And that's how we get water to the top of a 300-foot tree without a pump. But here's another question. Why is it when you go to the beach and you take off your shoes and you step on the sand and the sand is extremely hot, right? You're like, ooh, ooh, ah, ah, and you run to the water and suddenly you step in the water and the water is cool. Why is that? The exact same sun with the same radiation is hitting both the ocean water and the beach. Why is the ocean water not as hot as the sand? Here's why. As a photon of light, an energy particle, we'll talk more about that later when we get to energy. As a photon of light comes down from the sun, it actually hits that hydrogen bond between the two water molecules and it breaks it. And so when you break a bond, you release energy. So when that happens, then that energy is actually used to make another hydrogen bond, to form another hydrogen bond. And so these bonds are formed almost as quickly as they're disrupted. And so the heat energy is used up in the forming of these new bonds. And that's why the sand is much cooler than the ocean water. So the last concept that we're going to take a look at and explain that you see in your everyday life is why water floats when you actually freeze it. So you could take, as you know, water, put it in a tray, put it in the freezer, you freeze it, you get ice cubes, you put those ice cubes in a glass of water, and the ice cubes float. Why is that? It's the same water, right? Well, it turns out that when water is in its liquid state, then the water molecules can move about freely, allowing them to be closer to one another. Except when we freeze them, this crystalline structure actually begins to form, and it's keeping them slightly further apart. And therefore, they're less dense. So here, when they're in the liquid state, the water molecules have a higher density. They're closer together. Versus once they freeze, they're in this lattice-like structure and they're further apart. They're less dense. The less dense object is, then it's going to float, right? So that's why ice floats on water. So we looked at some of the reasons why water has these unique properties. And one term that you do want to remember is the fact that water is a good solvent. It's actually called the universal solvent, and it's because of that polarity that we talked about, that it can actually go ahead and pull different molecules apart and break those bonds just because of the strength of that polarity and the bonding that's happening. So when you hear that, what is the universal solvent? It's water. All right, thanks for watching. Make sure you review the 3 types of bonds that we talked about: covalent, ionic, and hydrogen bonds, and what the strength of each bond type is. Covalent is strong, ionic is strong, and hydrogen is weak. Make sure you review the basic parts of the atom and their charges: the protons, the neutrons, and the electrons. Remember, the protons are positive, the neutrons have no charge, and the electrons have a negative charge. And then review the unique properties of water, the fact that we have cohesiveness, the fact that it can actually, due to those hydrogen bonds, have surface tension, and the fact that it's a universal solvent. All right, have a great evening or day or afternoon or morning or whenever you're watching this video. I'll see you next time in the live chat. Bye, everybody.